Joining us from University of California at Santa Barbara in the USA. And we will hear about mesoscale organization in models of biological tissue. So let us see if the sharing works so far.
I hope so.
Yes, no, we see you now. Okay, yeah.
You should be able to see my screen and you see the presentation, right?
Perfect, yeah, everything is fine.
Okay, thank you. Thank you very much. And first of all, thanks for inviting me. I'm sorry I'm not able to be there. Trieste is such a great place. I hope to visit again sometime soon. I'm going to tell you, so we're changing topics quite a bit, I have to say, because what I'm going to do is tell you about some of our work on the behavior of dense sheets of biological tissue. And in particular, what I've been interested in is understanding the relation between the cellular structure and organization and the mechanical properties of the tissue. So the work I will talk about today was mostly done by an excellent student, Austin Hopkins, who actually was an undergraduate at Johns Hopkins and did take classes with Mark Robbins, got very good training there. And the former postdoc, Benjamin Lowe, who is now at the University of Edinburgh, and also in collaboration with Davide Marenduzzo and his students, Michael Cheng of the University of Edinburgh. And you will see, I hope, that actually friction does creep in the talk a little bit, because indeed these cells that I'll be talking about are sitting on a substrate and then crawling on the substrate, and therefore taking advantage of friction to do so. So given this audience is probably not necessarily familiar with cell mechanics, I will start with something really simple here. So here's an individual cell crawling on a substrate. As you can see, it performs a directed motion, and this occurs both in vitro and in vivo. The machinery for the motion of an individual cell is fairly well understood. Essentially involves the interplay of force generation in the cortex or cell cytoskeleton, which is a network of biopolymers and motor proteins, essentially are force generators in the system. So interplay between forces generated in the cortex and transmitted through focal adhesion proteins that allow the cell to attach kinetically, dynamically to the substrate. Given the size of cells, the motion inertia is generally negligible, and so the dynamics of the system is overdamped, and essentially the propulsion arises from the balance of active forces generated by the cell and substrate friction. Now, when you have many cells in a dense biological tissue, and in particular, I'm interested in epithelia, which are dense monolayers of tightly connected cells that line all our organs and actually surround the yolk of an embryo in morphogenesis. So this epithelia is a cartoon, so they always sit on a substrate. And if you take an image of the top surface, it looks like this. It sort of looks like a foam, meaning what the lines are here are the edges of the cells. And it really looks sort of like a tiling of the plane by a collection of irregular polygons. But the cells are active entities and move collectively in these dense tissues. And this motion, this collective motion, is important for a number of biological functions. Here is an example. This is wound healing, dense collections of cells moving collectively and coherently to fill in wounds, as you see here. And another example, of course, is morphogenesis. This is the embryo of a fruit fly. And there is this sheet of epithelial cells surrounding the yolk that rearranges itself. And what you see here is that in some sense, this tissue has to behave like a fluid to sort of flow to the right place at the right time. But once it gets there, it also has to be able to sustain shear stresses. So it acquires some elasticity. And so that raises the question in some sense of whether liquids, tissues are actually liquids or solids. Now, indeed, there has been a lot of work in recent years, both in the experiments, describing essentially tissue as materials near a jamming, unjamming, or liquid solid. I will use these terms a bit interchangeably. So near a jamming, unjamming transition. And of course, what you see here is again one of these epithelial tissue where cells are dividing. And so the system is getting denser and denser. And this kind of tissue is jamming through crowding, through increasing of density, which is not surprising, very much like what happens in collections of particles that can jam or undergo transition to a solid state at high density. More surprising is that living tissues can actually jam at constant density. So here, these are actually bronchial cells from human bronchial cells. I work from the lab of Jeff Fredberg at Harvard, and it's not as easy to see, but as the tissue mature and going from the top to the bottom image is about a week time, the cell's motion slows down appreciably and the tissue becomes jams. And actually the experimentalists counted the cells and the packing fraction is essentially remaining constant in this. What's happening here is that cells are essentially increasing the tension in the cell edges, and that leads to this kind of rigidification. Another example is actually this data from the lab of Giorgio Scita at the University of Milano, where what's happening here is that the top image, there is very little cell motion, so we call this a jam tissue. And then they actually overexpress a protein that in some sense enhances cell motility. And then the tissue unjams and it exhibits these large flowing motions. It becomes more like a fluid. So again, many ways for a tissue to regulate its rheology, to undergo sort of liquid-solid transitions, including a constant density. Now, so I told you a little bit about this. What I plan to do for the rest of the talk is tell you about some models we use to describe this kind of materials, to describe tissues as materials. And one important point that has come up in recent years is the notion that the fluidity of the tissue correlates with the shape of cells. And I will most of the time spend on telling you about a class of models that we call phase field models that essentially describe cells as motile and deformable particles with over-damped dynamics and show you how we can describe liquid-solid transitions tuned by cell motility and cell deformability. If I have time, I might tell you about some recent work on probing the mechanics of the tissue by what we call in silico active microrheology, so a simulation of what sort of a microrheology experiment. All right, so this is all two-dimensional. How do we model dense tissue layers? Well, there's been a lot of work on using particle models, describing cells as rigid particles, maybe even just disks, which are cell-propelled, including a motility. And of course, these models can actually capture liquid-solid transitions tuned by density and motility. But for these epithelial systems that are really very dense and where there are liquid-solid transitions at constant density, a perhaps more useful class of models is what are called vertex or Voronoi models that indeed describe the tissue as describe cells as polygons, and these polygons are tiling the plane, and each polygon has additionally a motility. And these models can actually capture, I'll tell you something about them very briefly, they can capture liquid-solid transition tuned by motility and by cell shape. And then most of the time I will spend on describing more recent work on phase field models that actually can additionally, where cells are deformable and motile, but one can in addition also vary the density. So essentially one is modeling here cells as deformable particles. I should say, I want to say from the outset that there has been a lot of recent work on using phase field model to model biological tissue by the group of Julia Yeomans at Oxford and Amin Dust Mohammadi in Copenhagen now. They take a slightly different view from ours, but that's actually they have done a lot of really very interesting work. So a few words on these active vertex Voronoi models. So the model essentially, as I said, describes the tissue as sort of a Voronoi tessellation of the plane with an energy. Each cell is characterized by an area and a perimeter, and there is an energy cost for deviations of area and perimeter from target values, A naught and P naught. And this target value contain essentially information about the properties, the mechanical properties of the tissue. For instance, the target perimeter is related to the cell edge tension. In addition, the cells are motile with a directed motility, and a lot of work done on this started by really my former colleague at Syracuse, Lisa Manny, and done by an excellent then postdoc, Max B, now at Northeastern, has led to the construction of this phase diagram in the plane of motility and target cell shape, so a measure of the shape the cells would like to have. And what I'm trying to show here is that this system exhibits a transition between a solid and the fluid state. The solid is a system where cells are sort of roundish, and they have a value of the cell shape defined as perimeter divided by square root of target area, which is smaller than this value 3.81, rather close to the value for pentagons. And they are caged. So we have both a structural and a dynamical metric for the transition. The structural one is in terms of cell shape, and the dynamical one is really obtained by measuring mean square displacement. And as you increase motility, they undergo a transition to a fluid state where cells are more elongated and they have kind of rougher shapes, and the dynamics, of course, and they're uncaged, they become like a fluid. This correlation that was seen here between cell shape and fluidity has actually been seen in a number of experiments. Here I'm showing some experiments. These are images from a developing fruit fly embryo that is sort of liquefying itself as time goes on and gets ready for invagination. And the experimentalists have actually measured this, what I call the cell shape, the ratio of perimeter to square root of area. And as the cell become more fluid-like and exchange neighbors, it is indeed increasing with time, establishing this kind of correlation between cell shape and fluidity. But as I mentioned, these models constrain the system to have constant density. This is really a tight network, and that's not always true. There are situations in which there are actually gaps between the cells. So more recently, we've been using these phase field models to describe the system, and that's what I would like to tell you about next. So we describe each cell as a phase field, a scalar field, which is 1 inside the cell and 0 outside the cell. And of course, for an isolated cell, the cell would be just round. There is an interface stiffness K that penalizes variations, and so sets the scale of the interfacial thickness and of the surface tension, which will represent the edge, the cell edge tension in the problem. And then we have a sort of a soft area constraint by varying this parameter lambda, which is a sort of compressibility in 2 dimensions. The tissues are really incompressible in 3D, but since we're just looking at a 2-dimensional model, they can actually change their area by changing their height. And this soft area constraint will allow us to have either monodispersed or polydispersed systems. Now, when we have to represent many cells, we use many interacting phase fields. So the free energy will be the sum of that single cell free energy. And we additionally include steric repulsion among the different cells. And our cells are motile. They perform persistent random walks, essentially move in a straight line at the speed v naught, we call it motility. And the direction of motion is randomized by orientational noise at a rate 1 over tau. Tau is the persistence time, which you can also describe by this very simple. So there is an equation for the dynamics of the phase field, which is advected by motility, and is not conserved. There is only this soft area constraint, and so the interactions come from a derivative of the free energy. Force balance is very simple. Friction of the cell is equal to the propulsion forces along the direction pi of cell polarization or motion. And this directed random walk can be sort of described as having some kind of a persistent noise with memory, given the way the persistence time tau. So the parameter in the models are, of course, the packing fraction, something we call rotational Peclet number, which is essentially the ratio of the persistence length of the single cell motion to the cell size R. And the parameter we call deformability, which is the ratio of the energy cost for two cells to overlap to the cell edge tension. And so it is a measure of whether it's favorable for cells to overlap or to deform. That's why we call it deformability. Now, if you simulate this model, what you find indeed is that the system exhibits solid-liquid transition tuned by motility and deformability. So a low motility, cells are actually form almost like a crystalline state for low deformability, and they're disordered, amorphous kind of glassy state, a high deformability. And as you increase motility, they melt into a liquid. And here are just some kind of videos of what your system looks like. These are very dense systems that we are considering, but there are occasional gaps. Now, this is just a schematic of the phase diagram. More quantitatively, we have quantified the transition by measuring both mean square displacement, and which of course will a long time become independent of time when in the solid where the particles are caged, and have a slope 1 in the liquid where the behavior is diffusive. And we have also measured structural quantities. We actually measure the structure factor, but also the bond orientational order, because this is a two-dimensional system. And it's been known that exotic type order in the bonds, the orientation of the bonds collecting a cell to its neighbors, has been seen in actually cell monolayers. And when we, now here is again the same phase diagram, where we have explicitly shown the quantities we've measured. So the fluidity is quantified in terms of the effective diffusion coefficient, the slope of the long time mean square displacement. And I think yellow and blue actually here are switched, sorry. The solid is where the effective coefficient is 0, and the liquid is where it's finite. And you also see a correlation with the bond orientational order, which is the background color in these images. But what is interesting is that if we look at this phase diagram a little bit more closely, we actually see that the melting is very different when the cells are highly deformable or when they are rather rigid. At high deformability, we find continuous solid-liquid transitions, and the fluidity indeed correlates with cell shape. So we have measured cell shape by essentially measuring the deformation tensor of our cells. And we have extracted this parameter that I mentioned in the context of a Voronoi model, the ratio of the perimeter to the square root of the area. And we find that indeed, just like in Voronoi models, the transition from solid to liquid occurs when this parameter is of order 3.81, and it correlates well with the transition at high, where the cells are deformable, at high deformability. At low deformability, we actually have the transition occurs like in 2 steps, and we have a sliver here where the system shows intermittency, that is quasi-periodic cycles of fluidifications, as apparent, for instance, from the behavior of the bond orientational order, that occasionally it's finite here for a while, and then you see occasionally dips when the system turns locally into a liquid, and then it solidifies again, and so on. So not quite a first order transition, not phase coexistence in space, but rather this sort of intermittency of the dynamics. I should say, what time did I start? How much time do I have? Okay.
Well, 10 minutes.
Okay, thank you.
Including discussion.
Yes, okay. And this intermittency same intermittency occurs when we analyze the topological defects. So the structural defects in our case corresponds to cells that are not six-fold coordinated, but that are 5- or 7-fold coordinated. So by performing a Delaunay triangulation of our system, we identify these topological defects. And we see that these cycles of fluidifications are indeed also associated with intermittent generation and annihilation of pairs of topological defects, suggesting indeed something like a two-step transition as familiar in two-dimensional systems. Now, I should say that in this region of low deformability, that our cells are sort of prefer to overlap than to deform. And indeed, we find that there is a correlation also with the location of the topological defects and cell overlap that we think of as a sort of a proxy for cell extrusion. Our model is two-dimensional, but in a real three-dimensional tissue, cells, of course, will not really overlap. They will actually extrude. They will be pushed out of the tissue and either start forming a second layer or sometime actually undergo apoptosis, die. And so we find a correlation between the overlap or cell extrusion with the topological defects, and in particular with a 5-fold defect, as shown here in these histograms. They show the occurrence of defects versus the local overlap. As you can see, the 5-fold defects are actually more strongly correlated with high overlap of the cells. And we understand this because essentially the 5-fold defects are a region where the compressive stresses are larger, while the 7-folds are the opposite, the region where the compressive stresses are lower. And so it kind of makes sense that this will be the region where cells tend to be extruded from the layer. Now, I will spend just 5 minutes telling you very briefly about work we are doing now on sort of trying to probe the mechanic of the tissue by what we call active microrheology. That is, we embed a bead in our model tissue, and we pull this bead a constant force and measure the probe velocity as a function of time. And of course, so this is a schematic of the response, the velocity as a function of the force. And if the bead starts moving right away, or we can have a situation where the bead starts moving right away, that is, there is zero threshold, or we might find a finite threshold for motion. And a finite threshold is sort of a suggestive of a yield stress material. And indeed, there has been experiments recently suggesting that tissue often behave like yield stress materials. Now, what we find when showing you here is the response velocity versus force for a system which is in the sort of highly deformable glassy state, where the tissue is quite compliant. You can actually, when you start pulling on your bead, it first responds by cells simply adjusting their shape around the bead. And as you can see, there are 3 regimes here. For very low forces, so first of all, there is a finite threshold force suggesting that this type of tissue does have a yield stress. For very low forces, the probe is caged and its velocity simply fluctuates around 0. For slightly larger forces near the depending threshold, the dynamics is stick-slip. And then eventually starts moving as sort of a steady pace with the slope approaching, this curve approaching a slope of 1, and very large fluctuations due to the motion of the surrounding cells. So there is a finite threshold, and this threshold does decrease with increasing deformability, as you again kind of would expect, because the tissue, the deformable tissue, is compliant and can adjust to the deformation induced by the probe. And here's a plot of this threshold as a function of deformability, which we think again of the threshold as sort of a microscopic measure of the local yield of our tissue. These rheological sort of simulated experiments confirm our observation previously of the difference in the melting between high and low deformability situation. And this difference is confirmed by looking at histogram of the instantaneous velocity of our probe in the two regions. At low deformability, actually at, let's see, in the region of low deformability, you find that especially near threshold, so these are histogram of the velocity, below threshold, the middle is just right above threshold and well above threshold. And as you can see, at low deformability, these histograms are much broader with fatter tails. We are actually hoping to be able to see sort of a bimodal distribution, which we could have described then as sort of a first order of plastic depinning in analogy with the driven disorder system, but that's not quite the case. We just see these really broad distributions, while when the system is highly deformable and compliant, the distributions are really remain very narrow. Since I believe I am almost out of time, I will actually skip a couple of slides and just go to the summary here. So I have been telling you about a model of biological tissue where we model the cells as deformable motile particles. And the model reveals two types of melting scenarios, which is sort of similar to what has been seen in of course, the models of rigid particles, as well as these Voronoi models, where when the cells are deformable, they sort of melt continuously by squeezing through their neighbors. When the cells are rigid, they melt here by overlapping, which we think of as a proxy for cell extrusion, with a two-step process and a region of intermittent fluidification cycles with the generation and annihilation of topological defects. We have established a correlation between the location of the defects in the cell packing and cell overlap, and in particular, we saw that cell extrusion is favored at 5-fold defects. And this is interesting to people who have followed this field, because there has been a lot of work on sort of correlating various biological functions even in cell packings with a variety of topological defects, either in the structure, as we have done here, or in the orientation of elongated cells. And we have looked at this in silico microreology that reveals the yield stress behavior of the tissue. And we are actually now trying to compare. There are recent experiments that actually are performing this type of microreology in real tissue, and we are trying to compare with those. Just as a teaser, I would like to just show mention something that we are doing right now. So I said before that these cells are moving on a substrate and they experience friction with the substrate. In addition, they also experience what I will call cell-cell friction, which you could, if you're thinking of the tissue as a continuum, you would call that probably viscosity. But what I call in cell-cell friction because it really arises from adhering proteins that mediate cell-cell adhesions, which are highly dynamical, that can come on and off on relevant time scales. And we're actually looking at the interplay between cell-substrate friction and the cell-cell friction in driving the organization of the tissue. And what we find is when cell-cell friction exceeds cell-substrate friction, our cells become elongated, as quantified by measuring the eigenvalues of the shape tensor, and acquire local nematic order as observed in many biological tissue. And as kind of shown here by these very brief movies, where the color here is the angle with the x-axis. And so this is something there has been a lot of work exploring mechanisms that may allow elongated cell to acquire orientational order or nematic order, as it's called, and associated defects, and various mechanisms have been proposed. And so we are exploring this one, which we think is directly relevant to systems when cells change to epithelia, when change, actually, so that's relevant to the situation where cells change their behavior from what is called mesenchymal. Essentially, they behave independently when cell-cell friction is not important, to epithelial, which is that the whole tissue behaves like a whole, like sort of a continuum elastic medium. So with that, I'll just go back to the slide to thank again the people who did the work, and I hope there might be some questions. Thank you.
Thank you very much for this overview and the modeling. So we have a question from Hartmut Löwen.
Hi, Hartmut.
Very nice talk. Thank you very much. So I have a question regarding the rigid cell case. So you were talking about the two-step process, the two-step melting. Is it really sharp? So what happens really in the thermodynamic limit? Is it sharp or is it a smooth crossover after all?
So you're talking about what we call cell-propelled particle or active Brownian particles? Or
about your
no, you're talking about my no, it's not sharp. That's why we would have liked to be able to say that there was really sharp transitions and perhaps something like a first order phase transition or something like that. We just see two crossovers. Yes, indeed.
Okay, thank you.
But the presence of this intermittent behavior is really localized to this narrow region between the two crossovers, and is very clear.
Okay, and one last question.
Sorry, maybe I didn't understand. In your phase field model, you impose a constant density of cell?
No, no. Each cell is a phase field, right? So what that means, let me go back here a second if I can find it quickly there. So what that means is that I have a phase field, which, this term allows us to have two values, 0 or 1, and I choose it. And so you can think of this, if you have a top view, if you just have one cell, the cell will be this round region. Inside the phase field is 1, outside is 0. It goes from 1 to 0 over a length scale, which is controlled by the stiffness.
So
okay.
Related to this, can you, I don't understand basically the meaning of your topological defect in this case. So do you have a conserved topological charge in your system?
No. So what we do to examine topological defects, we look at the center of the cells. We do a Voronoi tessellation of the plane. And then we identify, so, if you are into dimensions, you might, a crystalline system, your topological tessellation will give you a covering of the plane with hexagons. And what we find here is that the defects will be pentagons and heptagons. And that corresponds, that's actually what you do to examine melting in 2 dimensions of particles. You take the center of the particles, you do a Voronoi tessellation, and you identify particles that are not 6-fold coordinated in terms of the shape of the polygons in the Voronoi tessellation.
But the number is not conserved, basically. You can have they do not appear into pairs.
They do. They absolutely do, because they can only appear in pairs. You can never have, you must always have a 5 and a 7 coming in a pair. So there are defects in, so if these cells were all monodisperse and the density is high enough that they're closely packed, they will form actually an hexagonal lattice. And I don't think I have an image here, but you will see that the cells, because they're deformable, they get deformed and they form an hexagonal lattice. And if you were to do a Voronoi tessellation of that situation, which you don't even have to do because you already see the hexagonal lattice, you will see a covering of the plane with hexagons, okay? Defects correspond to either pentagons or heptagons, and yes, they will have to come in pairs. Topological charge is conserved. It's like two-dimensional melting, if you like.
Okay, thank you very much again for this clarifications and for the talk. And so we will move on now by thanking you for this talk. And
thank you very much.
Bye-bye, everyone online, and we will move to this online poster session, which will sort of be worked out within the next few minutes. And with that, I close this session here.
Stop the recording. Who is the recording?
Stop recording.